Hello everyone, welcome to part 2 of Learn Bioinformatics from the Scratch. This is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we will proceed towards part 2 and we will carry on part 1. In part 1 we have discussed in a brief details what does it mean the term sequence. So let's proceed. Okay, so today we will talk about sequence alignment. I'm sure you all have heard this name, like what is sequence alignment. So simply we can say that comparing two or more than two sequences, we call it sequence alignment. In sequence alignment, we can compare two, minimum of two, and we can compare more than two. It's up to you how many sequences you want to compare. Okay. Let's say we have two sequences, sequence 1 and sequence 2. Now, after watching the part first, you will be aware of this sign, the header sign. So, we have these two sequences. You can see that this is sequence 1, this is sequence 2. And there is a smaller dash in terms of bioinformatics or in terms of sequence alignment. We do not call it dash, we call it gap. So, what does this gap means? This gap is introduced during sequence alignment to improve the comparison, to improve the alignment. How? This is a question we will discuss in this session, and we will also learn the sequence alignment. So assume we have these two sequences, sequence 1 and sequence 2. These are nucleotide sequence you can see, and this is another nucleotide sequence. Now we have three scenario. The one is this gap. In bioinformatics we call it indel. Indel. N stands for insertion and del stands for deletion. So in comparison or in combination, the term is coined from the insertion plus deletion, so we call it indel. But why we do not call it indel insertion, or we cannot only call it deletion? The reason is just like of an example. For example, if I put half glass of water in front of you, so half of you may say that it is half filled, while the other half may say that it is half empty. So the same way, if I show you this alignment to all of you, some of you will answer me that in sequence 2, deletion has been take place. You can see there should be C, but there is no C character or nucleotides or base pair. So therefore it is deletion. But the other half, they may say that no, insertion took place. So we don't know this sequence was the real one or this sequence was the real one. So to kill the debate, like is it insertion or is it deletion in this case? So to kill that discussion or to kill that debate, the term indel represent this kind of scenario. So in this alignment, we are seeing a scenario of indel that explain insertion and deletion, and you can see here. Now, another scenario in this alignment, we will see matches and mismatches. For example, A matches exactly adenine from sequence 2. So this is match, thiamine, guanine, again adenine, thiamine, cytosine. Now, G and A, this is a mismatch. So this is this scenario. So in conclusion, sequence alignment is the comparison of two or more sequences, and we will face three kind of scenario: match, mismatch, and indel. I hope it is clear now. Now, there are two types of alignment based on the alignment procedure or based on the number of sequences. If you are performing alignment between two sequences, like I did here, then we will call it pairwise sequence alignment. You can see from the word pair. So pairwise sequence alignment. But if your alignment contains more than two sequences, let's say 3, 4, 5, 6, 7, 8, and so on. In that case, we will call it multiple sequence alignment. So, from now on, if your teacher asks you, you perform pairwise sequence alignment or multiple sequence alignment. So, just think of the sequences you compared. If you compared only two sequences, just say pairwise. If you compared more than two, just say multiple sequence alignment. Okay. Now, we are proceeding to the next slide. The next slide name is scoring scheme for alignment. You all are aware that once we align a data set or once we align some sequences or we are using some server just like Blast on the NCBI or some other programs like BioEdit or Mega. So these program always deduce some scores, and based on that score, they will tell you what two sequences are more related or dissimilar or not related. So, what is the scoring scheme or how this program scores the alignment? The score scheme is very complex, but as far as I assume or I suppose that most of my viewers watching this video, they are not ready for to just swallow the advanced level. So we will start from some basic. If there is a match case during the alignment, the program will give you positive score. If there is a mismatch case, the program will give a negative score. If there is gap introduction, program will give negative score. If there is gap extension, the program will give another negative score. Okay, what I mean from gap introduction, this here you can see a gap in this sequence. So this is a kind of gap introduction. So for this position, the program will give a negative score. But if in some scenario this gap is extended like dash, gap, 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 in that case, the program will give more negative score. Okay, so two negative scores for gap extension and gap introduction, and in bioinformatics we call it gap penalty and gap extension penalty. Okay, now let's not making things complex. Assume for match, program will give +1. For mismatch, the program will give -1. For gap introduction, the program will insert zero point 0.5 minus zero point 0.5, and for gap extension the program will produce -2, or let's say yes, okay, -2. Okay, now let's dissect these two alignment and you will learn something new. Let's say this is sequence 1, ATCGGAT, and this is sequence 2. Now let's calculate the score. A +1 because of the gap minus zero point 0.5, +1, +1, +1, +1, and because of the mismatch -1. If we combine this score, it will be 1, 2, 3, 4, 5, and then -1 and minus 1.5. So it will be, I said, okay, I mentioned 4.5, but I think it's 3.5, but that's okay. Okay, so the overall score will be 3.5 or 4.5. Now assume this alignment. Remember, the difference between these two alignment is the gap. Okay, you can read it: ATCGGAT, ATCGGAT. Now this one ACGGAA, ACGGAA. So in this alignment, you are not seeing any gap. It's just comparison, sequence 1 here and sequence 2 here. Now calculate the score. Plus 1, mismatch. Minus 1, mismatch. Minus 1, plus 1, minus 1, plus 1, minus 1. I think the score is minus 1. 1, 2, 3. And 1, 2, 3, 4. So minus 1. Now compare these two scenarios. The same sequences, two different alignments. Compare these two scenarios. In this case, if we assume that this result is the correct one, it means both these sequences are not related to each other because the score is in minus. But if we assume that this result is the correct one, then it means that these sequences are related to each other as compared to this result. So what is the difference between these two methods or between these two alignment? Because sequences are similar. The difference is the gap. This gap. If we remove this gap, then this T will be aligned for C and the whole this part will be shifted towards here, just like this. And in that case, we will believe that a particular sequence, particular set of sequences, they are not matching each other, but in reality they are matching each other. You can see. So it is just the importance of gaps, like how much gaps are important. But a question may comes in your mind that if gaps are so important, then why gap extension penalty? Why we give gap extension penalty? Because if you increase these gaps more than some level, so by this rule you can make any two sequences identical to each other or similar to each other. Let's say my one sequence is ABC and my another sequence is DEF. So you know ABC and DEF, both these sequences are completely different. But if we allow the use of gaps, then we will say ABC and then gap, 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 and then DEF, gap, gap, gap on the upper sequence. At this, by this rule, there will be some ABC in the first sequence later on, and maybe some CED in the second sequence. And that way, a time will come that you will say, okay, both of these sequences are related to each other, but in reality, they will not be related to each other. So that's why people are always saying X's of everything is bad. So gap, too many gaps will also impact the score to negative. So that's why gap extension penalty is there. There are different types of gaps, but we will not discuss it there because from these slides, I just want to give you a concrete idea how things are working. And then later on, when will we move to the advanced level, so it will help us to understand our results. We are not interested, or I'm not going to tell you each and every minute detail because I know you may not be the student of directly of the bioinformatics. You may be from microbiology, biochemistry, or botany, or whatever. So in that case, you don't need to to read each and every possibility of these things, but it's okay. So this was all about the importance of gaps, and this was all about how programs calculates the alignment scores. I hope you understand it. So let's move to the next slide. Okay, alignment amino acid versus nucleotide sequences. In our first part one, we talked about amino acid sequences or protein sequences, both are same thing, or nucleotide sequences. Okay, let's say this is a nucleotide sequence ATCG. When we perform alignment of the nucleotide sequence, like in this case, we have only two main scenario apart from the gap. Okay, miss a match case and mismatch case. Except that in case of the nucleotide, we have the third scenario of gap. But what about amino acid sequences? In amino acid sequences, we have 20 different types of amino acids. I mentioned seven of them: alanine, leucine, aspartic acid, glutamic acid, lysine, arginine, and valine. Okay, so we will also have a similar case here, like if the second sequence contain alanine A here, then it will be a match case. If it contains L here, it will be a mismatch case. But no, in case of the amino acid, things are little bit complicated. How? The mismatch case in case of amino acid, the mismatch case is interpreted into two other terms: mismatch but similar, mismatch not similar. What does it mean? I'm not making you confuse. So let's think about amino acid classification. Have you ever studied biochemistry or introduction to biochemistry in your course? So there was a topic in the chapter of proteins, introduction of classification of amino acids. What does that mean? We will imply that concept here. Okay, what is the classification of amino acid? First talk about this. According to the classification, some amino acid are positive charge. Let's say histidine, let's say lysine, let's say arginine. We call it basic amino acid. They contains positive charge. They contain positive charge. Okay. On the other hand, another group of amino acid, they contain negative charge, and we call it aspartic acid and glutamic acid. Now, there are some amino acids which are hydrophobic in nature. Let's say alanine, valine, leucine, isoleucine, and few more. I think seven or eight in total. They are hydrophobic amino acids. Some of them are polar amino acids. Polar amino acid means they contain both positive and negative charges. Or let's remind the concept of dipole-dipole from chemistry, dipole interactions, which has two poles, positive and negative. So it's okay. So the term polar means those amino acid which contains plus and minus in their R group. Okay. So if this is a classical case now. If sequence 1 is alanine, but sequence 2 here is alanine, again, the sequence 2 also contain alanine A in this position, it will be called perfect Karl match case. What if the sequence 2 contain valine, V, alanine and valine, V? In that case, they are not a match case because the sequence 1 has A, but the sequence 2 here, let me check if I have a, okay, here. Let's say this is sequence 1, this one. Now here we let's say this is sequence 2. So the sequence, this is sequence 1, okay? Now this is sequence 1. Here we have the sequence 2, so I'm going to draw it. So in sequence 2, let's say here we have A, so this is match case. Here we have, let's say, V. So now L lives in and V valine. This is not a match case. But is this a mismatch case? Yes. But which category? This is similar. Why? What I mean from similarity? What I mean from similar in case of amino acid or protein? From the similar, I mean that leucine and valine, they both are hydrophobic. They both have the similar properties. They both do not like interaction or interacting with water. So the property is same. So that's why this is a similar case. Although it is a mismatch, but it is a similar case. Okay, this concept will also help you later on if you are working on some sequence data or some mutation data. So you will see the different programs will tell you like this is the deleterious mutation or the damaging mutation, because in that case you will see, for example, if here is E, so E is glutamic acid, which is negative charge, but it has been changed, let's say R. In the mutant, it is R. So R is positive charge amino acid. And in this case, the program will tell you that this position contains the damaging mutation. So if E in one sequence and in the second sequence if the same position contains R, so it is a mismatch case but not similar case. You can say it is a mismatch case and this one case, not similar. But if L and V, L and A, L and I means isoleucine, they will be regarded as a mismatch case but similar. So in case of proteins, the same substitution rate or the same scoring scheme is not applied. If this is A and A, so let's say the program will give +1 score to this position. Okay, but if it is L and V, the program may give, let's say, plus 0.5, because it is not a match. It is not 5, actually it's not clicking, so it's I want to write 0.5, because this is not a match but it is not completely a mismatch. They belong to the same classification or the same group of amino acid, both are hydrophobic. But if it is this case, let's say E into R, so the program will give you a high negative score because it is a mismatch case with this category, not similar. So I hope you got my point that how the amino acid alignment is more complicated for the program. So to solve this issue, different algorithms are there, but in bioinformatics, two algorithms in protein alignment we are using. One is PAM and one is BLUESUM. So now you will, PAM stands for the point accepted mutation, or the BLUESUM stands for the block substitution matrix. You can Google it. But now you will have the answer if someone asks you why these matrices are not used for the nucleotide alignment. Because these matrices or these scores are calculated for protein or for amino acid cases. Why? Because the protein cases are little bit complicated. I explained in a little earlier. Okay, I hope I explained this slide. Now we are moving to the next slide. Okay, let me do it because I use this for the first time. Let me clear it. Okay, welcome back. So now what is alignment? Okay, in alignment, now we will talk about local versus global. You may heard the name like global alignment and local alignment. So alignment basically, as I told you, it's a comparison. If the comparison is made on the entire length of the sequence, we will call it global alignment. But if the comparison is made in the subsequence level or at the subsequence level, then we will call it local alignment. Let's say, what does it mean, the subsequence level? So this is, we here compare the overall sequences from here to here, from here to here. This is called global alignment, a classical example of global alignment. But what it means, what I mean from when the alignment is made at the subsequence level, we will call it local alignment. Okay, concentrate on this A, T, C, this is, let's say, a sequence 1 and this is a sequence 2. Now remember, if we made it alignment just like this, just keep sequence 1 and then keep another sequence and then present gap and blah 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 and calculate the score and then decide which one is related or which one is not, it's too easy, but it can lead you to a mistake. How? This is my sequence 1 and this is my sequence 2. If Let me tell you one trick. Both these sequences contain an interesting trick. The sequence this contain okay, this one. Okay, I will use this one. No, this one is okay. It contains these sequences TAT this box, and the sequence 2 also contains this box. Now, if you have studied some biology, so you will be aware that the TAT this sequence comes in the upstream level of a gene, and we call it the regulatory region. And this region is also called Tata box. And the importance of this region lies that the transcription factors recognizes the Tata box. Okay, now I know, or human knows, that these sequences are actually the sequences of regulatory region because it contains Tata boxes. But the computer or the alignment program don't know. So if the alignment program handles the alignment of this sequence globally, like started from here, match, not match, okay, insert a gap. So maybe this T will be matched here with some gaps or blah blah, whatever. So the result may come that both these sequences are related, or the result may come that both these sequences are not related. If the program says that both these sequences are related, that's great. But what if the program says that both these sequences are not related? That will be damaging because we know both these sequences are the sequences of regulatory region. Both they contain the Tata boxes, or maybe some other boxes, or maybe some other motifs. We know, okay, but the program says that they are not matching. Why? Because the program did it with the global strategy. What if the program first find out that is there any stretch of sequence, is there any patch of characters or bases which are exactly matching to each other. So yes, it has. So this is patch 1 and this is patch 2. They both are matching each other. So in that case, the program will first exactly, it will put this data here, and then the program will bring two gaps here and will bring CG or GC, let's say, okay, like this, CG, and then we'll make another gap here. And this part, ATTGC, will be aligned as per rule. But the program will also tell you that these two sequences are matching each other. The program will not skip these two sequences just randomly. They will treat it that these two sequences belong to the same class of regulatory sequences or whatever. Okay. So that's why local alignment is more powerful than global alignment. Now, so that's why I said that globally they will treat globally, and in local, first the matching patterns will be identified, and then uh, around those matching patterns, the other alignment will be performed. So if you see, the alignment mainly decide or it took the decision, the alignment program, based on this conserved motif or conserved patch or this same patch. So based of that, we will call it local alignment. Okay. Now you will understand that why BLAST is called basic local alignment search tool, because it is a tool, because it search for alignment against different databases, because it's perform alignment and it's basic level anyone can use. But originally the 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 phenomena that is following that is actually uh, the local phenomena or the local alignment. Okay, so let's Control Z to clear all this. Okay, so I hope you understand this slide as well. Drop your question if you have any in the comment section. Okay, now what alignment shows? You may have a question that why this topic is so important, what it shows. So let me tell you, sequence comparison, it shows you sequence comparison for relatedness or similarity or dissimilarity. But why similarity or finding similarity is very important? Another question. Remember, sequence group together in the same class of family. So if you have a sequence data and you are finding out if you put your sequence in that data and then perform alignment, then you can find out where your sequence is matching to which group of sequences. And it can help you in some cases. For example, recently the whole world has faced corona pandemic. And if you remember, as the virus emerged from the Wuhan province of China and then it spread to other countries. So there was a quick debate, like whether from where this virus came. And there were two reasons. Some people were saying that it came from bat coronavirus, and some say it came from pangolin coronavirus. But later on, the case was justified for bat coronavirus. You know how quickly and why so quickly it happened? Because the data set or the databases contain the sequences of thousands and millions of these different viruses. You can see NCBI virus database. So just scientists from different places, they just use BLAST or any other software and they found that the corona sequence which we are extracting from patients and sequencing, and then we are when we match it or when we blast it through alignment, it is matching through the bat coronavirus. It means, and then they confirm that yes, it came from the bat. So that was all the wonders performed by the majorly by the alignment procedures or sequence alignment. Another, from after performing multiple sequence alignment, we also can deduce or infer a phylogenetic tree or a phylogenetic relationships of kind of, we can comment on evolution. So that's another thing. Also, the alignment has a great deal of application in forensic science. Like if you got some sample from the criminal area and then you got some sample from the suspected people, and if there is any matching or whatever you can say, so once again, it's the alignment, the success of an alignment or the importance of an alignment. Okay, but in protein case, there is also another importance. Remember, in case of protein, we can predict the structure of the function of an unknown protein based on the alignment. Also, we can predict the structure of an unknown protein based on the alignment. We call it homology modeling. So, if I tell the name homology, then what is homology? So, we will discuss or we will proceed this to part 3, and we will talk about what is homology and what are all those things. So this video is getting longer. I don't want to continue it more. So it's enough for now. If you have any question, please drop your question. And thank you for watching me and supporting me. And I would suggest, if you like if you're liking my videos, if you're a teacher, please share it with your students, with your colleagues, with your fellows, and I'll be happy. This is the one good thing you can respond me back. Okay, thank you so much. Stay happy. Bye bye.